I think it's going to be completely different. So first off, I apologize for my Demi Moore voice. I usually don't sound this way. I've got some kind of a low-grade virus that has just decided to live on my vocal cords. So my energy is good. We'll see if my voice holds up for 2 hours. So my story starts in 1944. In September of that year, the tide had turned in Europe. The Allied forces had liberated France, Belgium, and southern Holland and made incursions into Germany itself. A massive Allied assault was planned to liberate northern Holland. It was named Operation Market Garden, and at that time it was the largest airborne attack ever staged. It took place between September 17th and 25th, 1944. It was a spectacular failure. Afterwards, Hitler punished the Dutch. The Dutch, of course, were allied with the Allies and had collaborated with them. Afterwards, Germany instituted a complete blockade, including all food and fuel shipments into what was then German-occupied Holland. The Dutch referred to the winter of 1944-45 as the Hunger Winter. Winter came early that year and was bitterly cold. Food stocks were rapidly depleted. As of October of 1944, 4.5 million people in and around Amsterdam began to starve. That lasted until May of the following year, May 1945, which is when Holland was finally liberated from the Germans. The Dutch people ate tulip bulbs and stripped their homes for firewood. Tens of thousands died from malnutrition and exposure for absolutely no reason. So it's hard for us to imagine what it must have been like for them. So the Dutch instituted a strict rationing program, and the ration was 450 calories, equivalent to 450 calories per person per day. So imagining, some of us in the room are parents, so it's, your inclination would be to, give all of your ration to your children or half of your ration to your children. At 450 calories, I mean, that's, a drink at Starbucks. Some of those are 450 calories. So it just wasn't feasible, and this lasted 8 months. So it's hard to imagine what it would be like to live through this incredibly difficult time if you're a parent or an adult generally, but particularly imagine if you're a child, and you're a child going through, your developmental processes living on that little food per day. This is a picture of a 16-year-old girl who survived the Hunger Winter. Her name was Audrey Hepburn. Audrey Hepburn suffered from ill health her entire life, which she attributed to living through the Hunger Winter. And the Hunger Winter also influenced what she did after her acting career was over, when she became a spokesperson and advocate for UNICEF. So that's an aside. That's really off topic here, but the point I'm trying to make is that the Hunger Winter was awful. And living through it, there was nothing to do. There was no one, rich, poor, no one had any food for 8 months if you lived in northern Holland. And there was incredible suffering at that time, but because this is the Dutch, detailed medical records continued to be compiled. What the hell did I just do? Whatever I did, I didn't want to do it. Come back. All right, sorry about that. And this provided a unique opportunity to study the effects of famine on human health. And so this is another aside. So curiously, during the famine, so general health decreased, but people who had celiac disease, their health, the symptoms from celiac disease actually improved. And at that time, no one had any idea. It had been described and it had been named, but no one had any idea what caused celiac disease. So an interesting thing happened in the spring of 1945 is the Swedish managed to smuggle in a shipment of wheat into Holland, and the priority was they turned the wheat into bread, and they gave the bread to the children. That was the first thing that they did. And the children who had celiac disease immediately relapsed. So this was the very first evidence that wheat germ gluten was associated with celiac disease. So this is the point in my lecture where I'm biting my tongue and doing everything possible not to tell you this nonsense about these gluten-free diets that everybody's on these days. So I'll say no more. I'll get back to my topic. So it was noted beyond this observation about celiac disease that babies born during the famine were small. So that's shown here. This is an actual graph from a paper published after World War II. So the babies, not surprisingly, the mother was malnourished, the fetus was malnourished. If the fetus was born during the famine, dramatic decrease in weight. However, babies that were conceived during the famine but were born afterwards were normal sized, which wasn't too surprising. As soon as the blockade was lifted, food was rushed into Holland. People ate a more normal diet, and the moms recovered and the babies also recovered, not too surprisingly quickly. So the interesting thing about this is the children born during and immediately after the Dutch hunger winter have been studied their entire lives. And so here's two papers. This is one published in 1972 in Science, and I don't know if you can read this. This is their description of what I just said, which is a slightly different take on it, but it's more or less what I just said. But this is a paper from 2006, so these folks continue to be studied. So this is when they were the people who survived, the babies who survived the hunger or were exposed to the hunger winter in utero were 19 years old, and here's when they're 59 years old. They continue to be studied. There's dozens, probably, no, I'm sure it's hundreds of papers on these populations. And there's been many interesting observations. So one is children whose mothers were malnourished only in the early pregnancy, that is the first trimester, had higher obesity rates. So when these kids were born, when they grew up and were adults, their obesity rates were higher. They also had the children, born to these mothers, had other health problems in their adulthood, including diabetes, which is probably related to the high obesity rates, but also some mental health problems such as schizophrenia and depression. So that's not entirely unexpected. I mean, the mother was malnourished, the fetus was malnourished. Okay, so that more or less makes sense, that there would be some long-lasting changes. What wasn't expected was when they looked at the grandchildren, and many of these same health problems, including low birth weight, low birth weight under normal circumstances. In the grandchildren, this was not expected. So this last bullet point is not from the hunger winter. This is from the second big highly cited thing, which was a Swedish famine. So this is far, far north Sweden, and this is an area back at the turn of the 19th to 20th century. That area was so remote that oftentimes, the glaciers would come in and they would get cut off from the rest of the world. So they routinely, these folks that lived way up there, why they chose to live up there, I don't know, but they were routinely exposed to famines. And these people have also been widely studied. So the one finding that fits with my story with the maternal transmission is that, this is shocking, that if the mom experienced famine at any point during pregnancy, that the grandchildren had decreased longevity, so their lives were shorter. So this fits with my story about maternal transmission. But the other really interesting thing from the Swedish study is the paternal transmission line, which we'll get to later in the talk, like in an hour. But there is if the male was exposed to a famine just before puberty, his children and grandchildren had decreased longevity as well, which is kind of shocking. It was just this very one period. If it was before that or after that, no effect. So the bottom line here is here's a lot of evidence, or at least some evidence, that suggests that things happen to you. They might be visited onto your children, grandchildren, and now there's even evidence for great-grandchildren. So these are intriguing in some ways, well, in many ways, troubling findings, but do they really reflect complex or even novel biological processes? So do these changes in offspring and grand offspring phenotypes, do they require genetic changes? Because, according to Mendelian genetics and Darwinian evolution, these things should not be happening, okay? So now it's, 2013, there's this buzzword epigenetics. We're not going to talk about epigenetics right now, but, do you need epigenetic changes, if you know what those are, to drive these changes in phenotype? Does the answer to this first question depend on which ones of the parents was exposed to the famine, or a toxin or drug or whatever? It doesn't have to be a famine. We're going to be talking about drugs of abuse today. Believe it or not, we are going to get to drugs of abuse, even though I started in World War II. But does it matter whether the mother or the father was exposed? I mean, does that tell you something about the underlying mechanisms based on your parent or grandparent? Which one was it? Okay, so this brings us to the weakest slide in the next 2 hours of my talk. I puzzled, if you look on Wikipedia, you do not find a good definition for intergenerational or transgenerational, so unfortunately you have to deal with mine, and they're not very good, but I'm going to explain them in the next 15 slides or so. But this is the best I could do. So intergenerational inheritance is the transmission of information across generations due to environmental effects on the gametes, which is sperms and eggs, or the fetus itself. Okay, so that's an intergenerational change. So it's, you know, it's what we were saying, it's not too surprising. The fetus was exposed, the mom was exposed, so was the fetus, okay? That the fetus would have problems, not a big shock, okay? The interesting thing for us as scientists are transgenerational changes. So these are these changes, you've changed the germline, okay? The parent or the grandparent, it can't be the parent, which we're going to talk in a second, but if your grandparent or your great-grandparent had some change, you know, if it's your great-grandparent, there has to, and it came down to you, there's got to be a change in the germline. And here's that word epigenetic again. So transgenerational inheritance is also known as epigenetic inheritance, since these offspring phenotypes are changed in the absence of DNA sequence, okay? So the point here is, I mean, you can get changes in phenotype that have to do with changes in the DNA sequence. These poor people that were exposed to the radiation in Japan a year or two ago, whenever that was, I mean, they are going to have higher rates of cancer and things like that because, you know, it's such a, you know, radiation causes changes in the sequence of your DNA, some of which are going to be regulators of cancer genes. So that's just one example. I mean, there's leukemia rates in that part of Japan are going to go up. Everybody knows that. Same thing happened after Hiroshima and Nagasaki. But famine doesn't do that. I mean, you know, radiation is a very specific insult. I mean, exposure to famine, no matter how bad it is not going to change your DNA sequence, okay? And that's the, you know, before, until very recently, that's the only way that we thought that phenotypes and offspring could be changed was through, you know, some process involving, you know, Darwinian evolution. Okay, so which one of these two things, and I've just given you the answer actually, intergenerational or transgenerational, which one of these applies to the grandchildren of women who conceived during the Dutch famine? Oh, I don't want to give you the answer. That was the answer. All right, which one is it? So if you're the grandchild of a woman who conceived during the Dutch famine and you have an altered phenotype, is that intergenerational or transgenerational? Why? So inter means you were somehow exposed, right? If you're, were the grandchildren exposed? Yes. It matters whether they're male or female, whether the grandchildren are male or female. That's a hint.
Their eggs were exposed.
Your eggs were exposed. So this is an example of, you know, obviously I stole this off the internet, and this is an example with smoking. So it matters if the fetus is female, and I did not know this until very recently, your eggs actually form in the first trimester. If you're a female fetus, all the eggs you're going to have for your entire life are there early in the first trimester. So if the mom is exposed to whatever, you know, famine or smoking or alcohol or whatever, the mom is exposed, the fetus is exposed, and the fetus's eggs are exposed. So that's the grandchildren, okay? If this fetus is a boy, there's no sperm. You don't start making sperm until you're hit puberty, right? And then even when you do hit puberty, you know, you don't have the same sperm your whole life. You know, us guys, we turn them over, whatever, every few days.
But you have the same stem cells.
Stem cells,
I don't think that answers the question. I don't think it does. Because everything I read says it doesn't matter. But I can't answer, other than that, I can't answer the question. I don't think that, but
I mean, I guess it has to do with, we're talking about epigenetic marks like methylation, I think, but the production of sperm from the stem cell resets them.
And that's not true. That's a good answer, and I would like to latch on to that, but that's not true either, which we're going to talk about in a little bit. So I think that the stem cells don't matter. I can't tell you right now on my feet why they don't. No one's ever asked me that question. But
most of the just
Well, I can't give you an answer until I think about it. And this emphasizes the fact that, as we're going to talk about later, I am neither a molecular biologist, and I'm sure as hell not a developmental biologist. So with those caveats in mind, this is the traditional model of how you get things into the germline. Okay, we have a caveat that we can't account for, but generally speaking, we think of this guy, the grandson, that this guy can be germline dependent. Okay, so if the father and the father or grandfather is exposed, or the mom or grandmother is exposed to whatever, this lightning bolt is some sort of environmental impact on the mom or the dad or the grandma or the grandpa. So in the F1 generation, in both cases, the fetus is seeing the same thing that the parents saw. Okay, either in the form of the fetus or the sperm for the dad. So on the paternal line, once you get to grandkids, so you've got one more step over here. If it's a girl here, that the eggs, this is an egg in that mom. Okay, so you've got to get out to F3 to know for certain that you've got a change in that germ line. Whereas with the paternal line, if you get out to F2, you can assume that whatever change you're seeing has to do with the change in the germ line. And this guy here being the outlier, being a little bit different. That if it's the grandson of a grandmother that had some sort of exposure, you can make the assumption that you have a germline change. Everybody with me? Okay, so I know you all are biologists, you all know about Darwin, so we're going to go through this really quickly, but I think it's important to at least to talk about where we come from historically and how we've gotten to where we are today in terms of this thing called epigenetic inheritance. So Darwin tells us that individuals less suited to the environment are less likely to survive and reproduce, and the opposite is change, and that drives natural selection. That's what we all learned when we were undergraduates. This slowly affected process, emphasis on slowly, takes place across millennia, results in population changes, changing to adapt to their environment, and it can even form new species. And so you guys all remember, the main thing that Darwin studied, and it's beautiful. Reading this book, I said to a group bigger than this one time, none of you have read Origin of Species, I said to that group, and one guy's like, yes, I did, I read the whole thing. Well, I tried. If you've read that thing cover to cover, you're better than me, because it's not well written. There's some brilliant stuff in there, but man, is it dense. And he goes on a million tangents. But he's got a lot of interesting stuff in there. And his observations with finches, so that's, he looked at the beaks of finches and showed that, on this little island that you had, some, finches, and he came up with this term niche. He said that the finches over here developed their beak because they're over in this niche and these over here. It was some really neat observational stuff. And back then that was what was in vogue was observational biology. And he made some really accurate and interesting observations. Then we've got this guy. Unfortunately, we have French people in here to tell me that I can't pronounce Jean-Baptiste de Lamarck, but this is Jean-Baptiste de Lamarck. And for all you French guys in the audience, I am going to resurrect Lamarck by the end of this talk, but first we're going to have to say some unkind things about him. So what Lamarck believed was that acquired characteristics are heritable, okay? That's the same notion that I just described to you with this epigenetic inheritance. But he came about it from, whereas Darwin's observations were really clever, Lamarck's really weren't. So there was two main things that Lamarck really focused on. So one of the things he noticed was that blacksmiths had big, well-defined muscles. And he noted that sons, I'm not being sexist, it's his words, not mine, that the sons of blacksmiths also tended to be blacksmiths and had big, well-defined muscles. Okay, so he thought, rather than the obvious, which, strikes us, why they both had big muscles, he thought that the guy being a blacksmith, then he had a son, and then the son had the big muscles because the dad had big muscles. That was his notion. And what he compared this to was basket weavers. So he made the same observation with basket weavers. Basket weavers did not have big, well-defined muscles, and the sons of basket weavers didn't have big, well-defined muscles. So this was Lamarck. The other famous example from Lamarck has to do with giraffes. And so his notion was, and we can think about this evolutionarily, but he thought it was just had to do with stretching of the neck. So he thought in one generation they were trying to get the leaves up high, and they would stretch their neck, and then the next generation of the neck stretchers would have a longer neck, and et cetera, across generations. Whereas Darwinian evolution says there's a natural, that some necks are going to be longer, some necks are going to be shorter, and the ones are going to get selected for. I mean, if it was little bushes in their environment, the short necks would prevail. Whereas if it was high trees in their environment, those guys would be more fit and would tend to reproduce. So now we know that that's correct. So this is the stretched necks is the Lamarckian idea. So one of the things we have to emphasize is, people thought Darwin was really interesting at the time, but it was Mendelian genetics, which, took a long time after Mendel actually described it, to come into vogue, and then, Darwinian evolution then really came into vogue because now we had a mechanism. So we biologists, even back in those days, back in the 1800s, we really like a mechanism to explain things. Otherwise, we just had a bunch of finches, on the Galapagos Islands. So now we had a mechanism. Fast forward to the 1950s, then we've got deeper, whenever, DNA, whenever all that stuff came to be. Wow, okay, now it all makes sense. We've got a mechanism of transmission, we understand genes, we understand chromosomes. So, it's not just Darwin alone. It's Darwin and, subsequently being shown to be right and having a mechanism. That's what led to the Darwin we all revere today. Poor Lamarck in comparison. So people were, they were really arguing about this in the mid to late 1800s, and Lamarck had his champions, plenty of them. So a lot of people, there were Lamarckians, there were neo-Lamarckians, and they were arguing with the Darwinians and all the other forms of evolution at the time. But our kids, a child could come up with experiments that would show that Lamarck was not really onto something. But back then, in 1891, there was this is an English scientist named August Weismann, and who was a big, famous scientist at the time. So he said, okay, I'm going to definitively answer this thing. So he removed the tails of mice across 5 generations, which Lamarckians would said, okay, the tail should get shorter and shorter and shorter. But it didn't, as we know. Why did he perform this specific experiment? Because the neo-Lamarckians claimed that some Jewish babies were being born without foreskins. I'm not making this stuff up. So that's why Weismann, performed that specific experiment. And so that's the one. If you read about Lamarck, that's the controversy raged, and it was Weismann. Everyone points to this experiment as being the one that definitively said Lamarck was wrong. But most of you probably when I was in school, those many years ago, the only reason Lamarck was brought up at all was to do what I just did, which was ridicule the man, okay? If he was brought up at all. Now you hear Lamarck a whole lot more, and it's because of this word epigenetic again, and I promise we'll get to epigenetics eventually. But this transgenerational epigenetic inheritance is, this should not happen. From a Darwinian perspective, any experiment that tried to show that there's some change across 2 or 3 generations in phenotype was doomed to failure. That then could go into the germline and be transmitted down through the generations, just like Darwinian evolution. So the first demonstration of this happened, as I say right there, in plants. So Linnaeus, who is the father of modern taxonomy, this Swedish fellow, was working in the mid-1700s, and he described the first natural morphological plant mutant in, again, 1749. And it was in something called toadflax. So toadflax is Linaria vulgaris, and there it is. That's the wild-type version, that's the wild-type version, that's the wild-type version, and that's the mutant. Okay, when you go down a path like this, at least if you're a guy like me, my brain starts working, and I'm like, okay, imagine I'm Linnaeus. So Linnaeus in 1749 undoubtedly knew more about plants than I'm ever going to know. I never even took botany when I was in school. But I imagine, like I've got all this knowledge. I took all sorts of biology. I understand how genetics works. If I was Linnaeus in 1745, how would I know that that was a mutant? And damned if I know. And I stopped myself before I actually found the answer to this question. So I don't know. But imagine what would you do? So now we know that that's a mutant of that because it's been sequenced. So they sequenced the genome of this thing, and they sequenced the genome of that thing, and with the exception of a couple of SNPs, they have exactly the same genome. Okay? Same DNA, radically different phenotype. Okay, so now we know that tells us it's a mutant. Obviously, Linnaeus was 250 years away from being able to do that experiment. So if I was Linnaeus, what would I do? I would probably breed this guy with that guy, okay? And if we ended up with 25, 50, 25, I would say, oh, okay, well, that follows Mendelian genetics. So it must be, you know, if these guys are similar, they must be fundamentally different and heritable. But Linnaeus, I mean, again, we were 200 years away from Mendel. I'm going to move on to the next slide. This is a really neat paper. So this is the paper from 1999. This paper is a pleasure to read. So it says right here, if I can read it, maybe I can read it on, no, I can't read it on there either. So, wow. Here we characterize a naturally occurring mutant of Linaria vulgaris, originally described more than 250 years ago by Linnaeus. Okay, so again, everyone agrees that Linnaeus characterized it as a mutant. I don't know how, but let's take it at face value. So it's not too surprising that these guys wrote a really nice paper, because they went to the trouble of going to the British Museum, which is where all Linnaeus' samples are now collected, and they found the sample of this thing called Peloria. The mutant is called Peloria. They found the actual sample that was picked by Linnaeus, took a photograph of it, and put it in their paper that they published in Nature. So thus, if one day I could do something so cool, that'll be very unlikely. So what they did is they showed, as we show up here, that they demonstrated first off that this is heritable. They're the guys who sequenced these. They sequenced that, and showed that other than a couple of SNPs, they were exactly the same genetically. But this was heritable. You could breed these guys. And they found the mechanism, which was a very specific change in methylation of this thing called, I don't know how they pronounce that, LCYC. So LCYC is a gene that has to do with the linearity of this plant, and it's highly methylated in the mutant. Okay, so it's turning, this gene gets turned off, and you get the mutant. So this was the first demonstration of transgenerational epigenetic inheritance. This goes through, I mean, the plant, it's easy to do. They did many, many generations, showing that if it shows up, it doesn't show up just randomly like you expect with a mutant. When the mutation happens, it then gets transmitted on through the plant germline, okay? And this was the first example, 1999. What about mammals? Okay, so I'll just tell you, and this gets back to what Jess said a minute ago, is people in the plant literature, I mean, that got into Nature. Okay, because folks in the plant literature didn't have as big a problem with this concept as the biologists do. Because as you said just a little while ago, and we're going to get to this in a second, maybe before I tell you the background, so I'll just give it to you. So it's long been appreciated, long been appreciated back to the 1970s when things like methylation and its influence on DNA was first described. So we know it influences phenotype. So if you have methylation of certain genes in your genome, you can get cancer, and we've known that for a long time. And it's the cancer biologists who have really driven the epigenetic field. Okay, that's my definition of epigenetics, and we'll get back to that in a minute. So this is the point that I'm trying to make here. So epigenetic modifications were thought to be cleared through passage of the germline in mammals. Okay, so that is any epigenetic change that you have in the parent cannot possibly get transmitted to the offspring, because all those methyl marks, as soon as the egg and the sperm meet, they all get wiped out. I mean, all the epigenetic marks are gone. And if that's not enough, it happens at least twice more during development, that they all just get wiped away, or at least that's what we thought. What's my next one? Okay, but now we know that it's not all of them. So 20 years ago, we thought all of them were wiped away. Now we know it's just almost all of them. And the almost word there is what's critical. So 1999 was a big year for transgenerational epigenetic inheritance, because that's also the year that Emma Whitelaw showed that it could also happen in mammals. And so she got it in a great journal. This was in Nature Genetics, but note, she didn't get it into Nature, because there was, I don't know Emma Whitelaw, I don't really know the story, but I assume that there was a lot of pushback on this idea, because again, even in 1999, this is not supposed to happen. Okay, so this was a radical hypothesis. So by all accounts, again, I've never met Emma Whitelaw. Everybody says she's a very pleasant Australian woman. and I'm not attacking her personally, but I'll tell you, I'm going to give you the bottom line of this paper. Don't read it. Just trust me. It is dense. It is really hard reading. And you're going to fall asleep in the next 3 minutes, me trying to explain it to you. And I've tried to boil it down. I've done my best. But she studied this thing called the Agouti mouse. So Agouti mouse, so it's not, normally it's not a mutant, okay? These are mice that are found in South America. They're these mottled little devils, and the geneticists have known about them for a long time and have studied them, again, since the 70s, and they know all this stuff about Agouti mice, okay? So that's before we started genetically manipulating these they started genetically manipulating these little guys. So this normal Agouti mouse has hair that's banded. So it's black at the tip, it's yellow in the middle, and it's black at the base, each individual hair. Okay, and that has to do with the gene being turned on, turned off, turned back on again, believe it or not. And the Agouti gene is responsible for the yellow. Okay, so the yellow part that's in the middle, that's the Agouti gene. And these are genetic mice. So these the A stands for Agouti, VY/lowercase a, they have the Agouti gene switched on permanently. If the Agouti gene is switched on permanently, okay, so genetically, they should have yellow hair. This is done in a laboratory. They all have the same food. They all are raised in the same rooms. You know what I mean? So their environment's all the same. So they should all have, all these animals, these guys, these C57, this on that background, they should all have yellow hair. But they don't, okay? We're not to transgenerational yet. This is just describing the Agouti mice. All right, brace yourselves. These mice have an intracisternal A particle retrotransposon that codes for an RNA. It's not a microRNA. It's a non-coding RNA, which keeps the Agouti gene turned on continuously that results in yellow hair. Okay? So another aside here, who knows where the retrotransposon was discovered and described? Take a wild guess. Cold Spring Harbor. Does anybody know who did it? Barbara McClintock, very good. So Barbara McClintock, who worked here in the mid-40s, I don't know how long she worked here, but this work was, the original observation was in the mid-40s. So it's pretty cool that Cold Spring Harbor, funded this woman in science. It's not easy to be a woman in science now. It had been nearly impossible in the 1940s. But she made these observations, and she studied maize. So it was actually, again, plants, where she described, they're now called retrotransposons. I think a transposon and a retrotransposon are the same thing. As far as I know, they're the same thing. She called it a transposon. She published her findings, and I'm not going to get into what exactly it is. They're jumping genes. She published it in the early 50s, and nobody cared. And so again, the world took 15 years for the world to catch up to Barbara McClintock. And they finally, in the 1960s, they showed that these retrotransposons were in mammals, that they were critically important for gene transcription. And in 1983, she won the Nobel Prize. So that's a happy story. All right, so I'm not going to get into all that. So the point is about these Agouti mice that methylation of this retrotransposon DNA in these mice acts as kind of a dimmer switch. So if you have unmethylated animals, this retrotransposon does its thing, and we get yellow mice. If it's completely methylated, we turn off that gene, and we get the brown mice. And then if the level of methylation is somewhere in between, you get these mottled mice. Okay, so some are, you get some brown hairs and some yellow, some of the banded hairs and some of the yellow hairs. We're still not to transgenerational yet. We're just describing the Agouti mouse. But this is a classical case of an epigenetic modification because these guys have exactly the same genome, exactly the same environment, different hair color. Okay, so this is a classic case of what we would now call an epigenetic transgenerational modification. But we had to have Emma Whitelaw to show us this. So again, background for Emma Whitelaw. So to emphasize the point that these Agouti mice have been studied since the 70s. All these things, including the methylation marks, have been known. That was discovered in the 80s. And they knew, moreover, that it was derived from the maternal line. So it was a maternal influence, not a paternal influence. And what the folks in the field had assumed, because no one was thinking about epigenetic modifications other than methylation, so epigenetic meaning a transgenerational change in the germline, not a methylation change. So it was assumed that these metabolic differences in the intrauterine environment influenced, the level of methylation. So even though the environment seemed very similar, that there was some variation amongst the moms, they thought that must be it. Really there was nothing else it could be as far as these scientists were concerned. So one of the things Whitelaw did, and oh God, shoot me if I have to go look through all these cross-gestational things, finally I just took her word for it. They took the fetus out of this one and put it in that one and this one and that one and did all these crosses. And so I'm taking Emma's word for it that this couldn't, the bottom line of all these experiments was that couldn't possibly be true. So what could it be? So then we get to epigenetics. They thought that the maternal effect might be due to the persistence of epigenetic modifications through meiosis. So again, they're tilting at this windmill. That's because this is not supposed to happen in mammals. So they showed that this Agouti retrotransposon, methylation, was erased in the male germline. So it's not this phenotype, they knew it wasn't coming from the male germline. The male germline, all those methyl marks came off just like they were supposed to, but not in the female line. So the methylation was incompletely erased and led to the phenotype. And this was a big deal and remains a big deal. Because it was the first example of transgenerational epigenetic inheritance in a mammal. In other words, our friend Lamarck lives. So now, for the French folks in the audience, I'm going to resurrect Lamarck. I'm going to say some nice things after saying something. Nobody ever says anything nice about Lamarck. So I'm going to say a few nice things about Lamarck. So again, I want to emphasize this point that Darwin was great. He was appreciated in his lifetime. But if not for Mendel and Watson and Crick, what institution did Jim Watson head for 35 years? Hint, it's Cold Spring Harbor. Until he said some really, really stupid things in 2007 and got himself fired. He's not the director, though. So my Jim Watson story is Jim Watson went to the Indiana University, which is where I got my PhD. And so when I was in graduate school, they invited him back. This was in early 90s. It was 91 or 92. So he was an older guy, but he wasn't that old. And they invited him back. They put him, his talk was going to be in the biggest, the place that had the most, it was actually the auditorium, like 2,500 seats. The place was jammed, completely jammed. I mean, I was there. I went to see, the great Jim Watson. So Watson gets up there and starts rambling around, telling all these stories, and he was the head of the Human Genome Project at the time. The head of the Human Genome Project, and the guy was completely senile. Back then it was slide carousels. He never clicked onto his first slide. He told us how pretty all the secretaries were at the whole, I'm not making this stuff up. It was nuts, and he rambled on. And finally someone got up and took that, Nobel Prize winning Jim Watson, they gave him the hook, got him off the stage. It was so bad. And so, when he said that stuff in 2007 about race, I was not surprised. The guy, in front of all of us, talk about your heroes going down in flames. It was sad. Anyway, or whatever. But I lived. I moved on. So the point here is that, so Darwin really needed Jim Watson and Crick and Mendel to put all of his stuff in a context. And so now today we talk about Lamarck because epigenetics, which I promise we are going to talk about epigenetics in a minute, provides a mechanism for Lamarckian transmission of an acquired characteristic. So although some of these ideas were clearly wrong, so we know that we can have acquired characteristics go through the germline, but stretched giraffe necks doesn't tell us anything about it. So he was on the right idea, but whereas Darwin, very meticulously described all these things that he saw that were correct, Lamarck not so much. But what he was known for at the time is he was a highly, highly respected taxonomist. And indeed, he named over 1,600 species, including Aplysia, that, there's Eric Kandel books in the room back there, the thing that he and many other people study. So that was actually named. Lamarck was the guy who called it Aplysia originally. He also coined the term evolution. He was the first guy that, that came up with this term. And indeed, even today, Lamarck is credited with at least constructing a coherent theoretical framework. I mean, many of the aspects, the details are wrong, but at the time he was at least thinking about these issues. I mean, he got some fundamental things wrong, but even today, people point to this and say this was an important contribution. And none other than Darwin, in his own time, I love this quote, praised Lamarck for the eminent service of arousing attention to the probability of all change in the organic world being the result of law, not miraculous interposition. So that's a cool quote. All right, so we've spent a lot of time in this first part of the talk talking about genetic versus epigenetic inheritance, and I keep coming back to this word epigenetic, which I kind of sort of defined, but not really. Kind of told you, I'll get there in a second. Told you some things about epigenetics, and that's all great. But what the heck is epigenetics exactly? And that's what I'm going to explain in the next part of the talk.
So did Lamarck ever provide an example or an observation that can be explained, like not blacksmiths or giraffes, but actually can be explained by what we think of as epigenetics?
No.
Yeah.
Sorry, French guys. So now we're going to talk about epigenetics, but any questions about anything so far? So here's the point in the talk where I get to re-emphasize the fact that, and maybe this will be instructive, it's either instructive for you guys or I'm a cautionary tale. You can take whatever you want from what I'm about to tell you. So when I went to graduate school, I studied neurochemistry. I did a postdoc. I studied neurochemistry. So I did voltammetry. I did microdialysis. I haven't done either one. I haven't done microdialysis in 15 years. I haven't done voltammetry in even longer than that. So whatever you're doing right now, you think that's what you're going to be doing. I don't do any of that stuff anymore. And I was not trained to do anything molecular. Nothing at all. So when I was a postdoc, I started dabbling back in the days when PCR was hard as hell to do. We cast our own gels. So I did some of that when I was a postdoc. But it was, like at the very end of my postdoc, I was like, well, the guys over there down the hall are doing it. It looks cool, so maybe I'll learn it. But that was the extent of it. So, but now I don't do any of the stuff that I was trained to do. So, I mean, which may be stupid, but I'm still standing. So, take that for what you will. But, I think that, and not to say that the other is not, there's people that have done the same technique their whole life and, evolved, the technique has evolved with them and they've improved it. I'm not saying that's the wrong way to go, but that's not the way I went. And now I'm going to stand up in front of you and try to explain what epigenetics is to you. And maybe I'm the correct person to explain it to you. Now, some of you out there are molecular biologists, and please don't have mercy when you ask questions in a few minutes. But maybe I'm a good person to explain this because this is my second grade level of epigenetics, which, for those of you who are uninitiated, maybe that'll be a good thing. So now we're actually going to define what epigenetics is. So epigenetics, as I've hinted many times so far, are changes in phenotype caused by mechanisms other than changes in the associated DNA sequence. Okay? So the DNA is all the same, but the phenotype changes. So there's got to be some extra or epigenetic change that drives these changes in phenotype. So going back to my education being woefully inadequate, I mean, we had to have known this when I was in college, that there are nearly 2 meters of DNA packed into the nucleus. Nobody told me that. I didn't know that until just a few years ago. That's a lot. That is a big stretch of DNA in every little tiny nucleus. And the diameter of a neuron, I mean, there's all different sorts of cell types in the body, of course, but the neurons are pretty small, and their nucleus is, in the 1 to 2 micron range. 2 meters of DNA in 1 to 2 microns. How the heck does that work? How does it fit? Well, DNA is very thin, so like a width of around 2 nanometers. I mean, that's really thin, but still, you've got 2 meters of something that's very thin. Moreover, the DNA is really tightly wrapped around these proteins called histones. All right, so here you've got the DNA unwinding, and in this example, the histone is a ball. So you've got all these little ballies, and you can see how tightly the DNA is wrapped in there. So I mean, the DNA is wrapped super tightly around these histones inside the nucleus. And so here's another buzzword. So a key word here is the nucleosome. So the nucleosome is a nucleosome. It is the DNA which is wrapped twice around a histone. So in this case, it's a disc disc ball, whatever. I mean, this is artistic license for whoever made these things, and I stole them off of the internet. Okay, so the nucleosome is the combination of histones and the associated DNA. There are exactly 146 base pairs of DNA are wrapped almost twice around an octamer of histone. So this histone is actually 8 histones, okay? So there are combinations of H1, H2A, H2B, H3, and H4, and I've highlighted H3 because that's the one I'm going to talk about later in the talk. And actually H1 is not one of them, I just misspoke. So this octamer of histones is some combination of those guys, whereas histone H1 is a linker protein, so it's not actually part of that ball or disc. Okay? All right, so here's our terms so far. A histone is a protein found in the nucleus that forms the spool that DNA wraps around. A nucleosome is a single section of DNA wrapped around a collection of 8 histones. And chromatin, you can see it's all in there like spaghetti in the nucleus, is all DNA and all histones in the nucleus. Everybody with me so far? Like I said, 2nd grade level. All right, so next concept. We've got heterochromatin versus euchromatin. So heterochromatin is euchromatin. That's this. So the chromatin is hetero or closed, and transcription is silenced. So the point of all this is this chromatin is so tightly wrapped in the heterochromatin state, so it's got to be that tightly wrapped to fit in the nucleus, okay? And in most of our nuclei in our bodies, the vast majority of the DNA is in that state, okay? But the thing about that state is that's good for packaging, that's terrible for transcription. This DNA is so tightly wound in there that the transcription factors can't get in there to do their work, okay? In order for the transcription factors to have access to the DNA, you've got to unwind it, okay? And when you unwind it, it's called euchromatin, which is open chromatin. The transcription factors have access to the DNA, and you're ready for business. So heterochromatin, here it says transcriptionally silent. Epigenetic events like DNA methylation is one that we've talked about in the first part of the talk, but then also we're going to really focus on histone modifications in the next part of the talk. So the point being here is this is the DNA, and there are things called CPG islands. Who knows what a CPG is? What does CPG stand for? What's the P stand for? It's real. You're not going to believe it if you don't know. Proximal. C proximal to G. So these CPG islands are just C's and G's. They're next to each other. And if they're next to each other, so for whatever reason in biology, that's the thing that gets methylated. The C's and the G's have to be next to each other, and then the C gets methylated. And that's just one example. So that's DNA methylation. So that's an example. I think I talk about this in a minute. But you can imagine, and I alluded to this just a second ago, so during development, that's where a lot of these epigenetic, quote unquote, changes take place. The developmental biologists are the ones who identified them, because obviously, you know, you've got every cell in your body has all the genetic material capable of producing anything. You know, it's producing a liver cell, a brain cell, a toenail cell, you know, whatever. I don't know, toenail cell? Yeah, or whatever. Every cell type in your body, right? All of your neuron cells, your neurons, shouldn't be producing liver cells, okay? And, you know, our eye cells shouldn't be producing teeth. So we got to turn all those genes off. And that's the way our body does it. It methylates the DNA. So you turn off the DNA, and that causes all those genes we don't need in a neuron to be turned off. Yeah?
First grade level question.
Okay, maybe I'll be able to answer it.
Is there any sort of like sequence specificity to the way that the histones interact with the DNA, or you know, the way they wrap around? Is that kind of dependent at all on the sequence, or at least its charge properties, or? You know,
so the charge properties. So it's
it interacts with maybe the backbone phosphates or something like that. I don't.
So it's the charge that. So my first grade understanding. So it's definitely the electrostatic charges that cause it to be in one conformation or the other conformation. But it's, you know, it's just, you know, it's all this DNA. It's like every 146 base pairs. You know, I mean, it's you know, they're all random, and I mean, there's a lot of histones. So it can't, I mean, there's nothing that, all these sequences are different. So I think in answer to your question, to my knowledge, someone correct me if I'm wrong, is that there's no specific way that they link to each other than in a general electrostatic way, that, you know, the positive and negative charges, that these processes like acetylation and methylation cause the charges to be either there or not. And that's what causes them to wind and unwind.
That's why the H1 histone is so important. That's the linker which is like put all them together. Like, I don't know if you go on the picture before.
So the little yellow thing on the side.
I
Yeah.
remember seeing
Yeah.
that one, yeah. So,
Stabilize the whole core.
I figured it has to be very promiscuous. So.
So I think promiscuous is the correct word. So, but that's DNA methylation. So DNA methylation during development is critically important. That's how we get the different cells in our body, and they're just turned off forever. So during development, that gene gets turned off. You know, if it's a liver gene in a neuron, it's never going to come on unless you get some, you know, weird mutation like, you know, exposure to radioactivity, like we said. But the other process is, notice this guy here. So this is a really cool picture. So that's the DNA, and that's a collection of histones there. I mean, so that gives you an idea of what it looks like. I mean, that's it. I mean, at least, you know, a stylized example of it. So like H3 is here in purple, and this is, that's the octamer of histones right there with the DNA wrapped around it. But the critical thing here is looking, again, this is H3, which is the one we're going to come back to. You've got these little tails sticking out beyond the DNA, and we can modify those tails. And these same epigenetic processes can take place on these tails, okay, like methylation, acetylation, etc. So here we have, so DNA methylation is one form, one epigenetic mark that can, you know, turn on or turn off the DNA. But then you can also modify the histones in a similar way to produce a similar effect. So, for example, you can methylate the histones as well. And so, again, it does the same thing. So you end up with a heterochromatic form, and that suppresses gene transcription. And so this is the point that I've been making about, you know, turning genes on and off. So we've been talking about methylation a lot, and I've been comparing and contrasting that with acetylation, but there's a whole lot of other processes. So all of these are epigenetic marks if they're associated with the histones. So we're going to focus on these two, but there's also all these guys as well. And so here's our CPG island. So that's a C proximal to a G, and that's got a methyl group on it. Okay. So that's just an example. If we methylate, you know, histone 3, for example, it's going to cause the you know, the heterochromin form, and it's going to shut down transcription. Okay, so the two that we're going to focus on are the two that are, methylation is by far the most studied of all these epigenetic marks. And again, for the 19th time, it silences transcription. And the other process we're going to talk about is acetylation, and that opens up the chromatin and causes the active form. All right, so why are we talking about this? So the first thing to notice is some of these genetic diseases actually have come from an epigenetic component. So the most widely studied is something called Rett syndrome, and there's actually a Rett syndrome foundation now that they support research. And Rett syndrome is an autism-like disorder that is, there are some cases in boys, but it's predominantly in little girls, and it is beyond severe. So I mean, autism-like doesn't even touch it. So they have microcephaly, they have all sorts of changes in behavior. They're most known, I mean, some of these little girls never are able to talk. I mean, there are severe developmental problems. They die very young, but those that, you know, they have all sorts of seizures and problems, but the one behavior that most of them show is this like hand-wringing behavior. So, I mean, it's incredibly sad. I've seen videos. I mean, it's heartbreaking to see these poor little girls. And this is a mutation in MECP2, which you probably heard of, which is X-linked. And MECP2 is methyl CpG binding protein 2, and it's a gene that alters chromatin methylation. So
Is it chromatin methylation or DNA methylation?
I thought it was chromatin. Is it DNA?
I think it's DNA.
Good to know. So that's
Also
wrong.
binds hydroxymethyl C.
So it influences methylation. I think we can all agree on that. Another example is Rubinstein-Taybi syndrome, which I had never heard of. But so MECP2 is studied a lot. CREB, you all have heard of CREB and addiction. Eric's going to be talking, I'm sure, about that tomorrow. So these kids have, this is both boys and girls. I think it's actually more in boys. They have a sloped forehead. They have really strange phenotypes. They're small in stature. They have wide thumbs. Again, you know, their developmental problems. You know, their IQs are extremely low. So, so these are two examples of, and this is an acetylation linked. So we're going to talk today about MECP2 and methylation and CREB and acetylation. So those are quote-unquote genetic diseases that are actually epigenetic diseases. But what about environmental diseases? So we talked about cancer previously. So, you know, if you have DNA methylation causing the loss of expression of a tumor suppressor gene, okay, if that happens, that you're going to end up with cancer. And so that's why, and this is, I don't know anything about cancer, but I know that epigenetic processes are by far the best work out in the field of cancer. And so then the question, if all of this is true, and we can have changes, these epigenetic quote-unquote changes and all these other things, well, what about addiction? And that's what we're going to talk about in the third half of our talk, which we will do after a 10-minute break.
Sounds good.
Thanks. Yeah, that was stupid. Then everybody in my lab got to shoot it. But if I got them, they had to come on. Good call. All right. All right, we're going to talk about the sins of the fathers. And we're going to talk about forgiving the sins of the fathers. And I'm Chris Pierce, and I clearly have way too much time to fool with PowerPoint. And that's where I work. Okay, so before we get in, so believe it or not, we're actually going to talk about addiction today. And so before we get into the epigenetics of the epigenetic translation of addiction phenotypes, let's go through a brief history of cocaine. So here's some more fun and games to keep us awake at this late time of the day and make us not think about going to the pool. So a brief history of cocaine. So while it might appear that cocaine use and abuse exploded on the scene both in this country and worldwide in the 1970s, the history of the drug dates back at least 2,000 years to the Incan civilization in Peru. And this is where the coca plant is indigenous. So it's very curious that even in 2013, the only places that that cocaine is cultivated are in these mountainous regions in Peru, Colombia, and Bolivia. And so one of the reasons that it's so hard to eradicate is because these are very remote areas, again, way up in the mountains. And these must be very interesting microclimates because, you know, if you could grow cocaine in Seattle, people would be growing cocaine in Seattle, right? So still, the vast, vast majority of it comes from these few regions of South America. And that's the coca plant, and it's the leaves of the coca plant is where we get cocaine. So the Incas clearly appreciated the pharmacological properties of the coca plant and that they claimed that God's angels presented man with a coca leaf to satisfy the hungry, provide the weary and fainting with new vigor, and cause the unhappy to forget their miseries. So this was written over 2,000 years ago and is actually quite, you know, an apt description of the behavioral effects of cocaine. So in the Andes, both historically and contemporarily, coca leaves are chewed or brewed into tea. So I've never been to Machu Picchu personally, but I've been giving some version of this lecture to medical students for like 15 years now, and these medical students, they go to Machu Picchu, and they come back, and I've got a whole drawer full of these things now. And I'm like, you know, it's like, you know this has got cocaine in it, right? And you got on the airplane with it, right? But evidently, if you go at Machu Picchu, if you order tea, you better specify non-coca, because otherwise you're going to get a little more bang than you were expecting. And my thing is that, I mean, you know, I drink alcohol, you know, I'm no saint, but for some strange reason I have never tried cocaine in my entire life, even though I've been studying it in my entire career. So I haven't brewed this stuff up just because they would break my record of never taking cocaine, but evidently it's really bitter. So, and we're going to come back to that in a minute. So, and they've been doing this for a long time. So this is a woodcut, and the Incas thought that the coca plant was a gift from the sun god. So this is the sun god presenting humankind with with coca in the form of coca tea. So they've been drinking coca tea for a long time, and they also chew it. So this is a jar where they put lye into the jar, and what they do, for hundreds of years, is they take the cocaine and mix it with lye, and they stick it and they chew it, and that causes I guess alkalinization, causes it to be absorbed better. And so this is just contemporary Peru. I mean, this is all coca leaves being sold in the market. Again, I drink alcohol. My bartender is Peruvian, and he says, when he goes, his grandma's got a coca plant in the backyard. They just go and they pick off the coca leaves, stick them in there, and chew them. But the thing about it, and the reason, so we're going to come back to why the coca plant didn't catch on in European society until very, very recently, is because you got to chew a lot of these things. I mean, you got to have a big mouthful. I mean, you can see this little lady here. I mean, that's a lot of cocaine in there. And you got to chew it for hours to get any sort of pharmacological effect. Of course, bioavailability of cocaine is really bad. It's like 15%. But I mean, see the quid that this guy's got. So this guy's a tin miner. So a lot of the down in South America, in those regions where the coca plant grows, up in the mountains, they also have a lot of mines. So tin mines, copper mines. You remember the Chilean miners? So those guys like that, they chew this stuff all day, and that causes two things. One, they're working at high altitude, and that allows them to work at high altitude. And the conquistadors, back when they took over South America, they enslaved all these indigenous people, and they forced them to chew the coca plant so they would work harder. So they can work hard at high altitudes, and they also don't eat. So it suppresses their appetite. So they can just be down in the mine, not worrying about anything but mining. So the coca plant was first introduced into Europe in 1533 by the Spanish explorer, and I'm going to say explorer. I mean, I'm not going to make any value judgments. Pizarro was not a nice guy, but he's the guy who brought the coca plant back to Europe. However, the drug didn't come into widespread use in Europe until the late 1800s, and this coincided with the isolation and purification of cocaine by the German chemist Albert Niemann in 1860. So again, the coca plant came to Europe. You can't grow it there again, so I mean, it's got to come all the way across the Atlantic Ocean to get there. It dries out if it's not on a plant. The plants don't like going across the ocean for a few months. So first off, just getting it there was problem enough. And once it got there, you brew up tea. Well, the tea was bitter, and Indian tea was available hundreds of years ago in Europe. And they're like, well, we'd rather do the Indian tea. And they're not going to chew it. Wasn't part of their culture. So, the Europeans weren't going to chew the coca leaves for hours to get any sort of effect. So there was a big shrug, essentially. So they knew about it for hundreds of years until Albert Niemann came along. So I don't know if any of you are graduate students, but Albert Niemann was a German graduate student. His PhD thesis project was figure out what it is in the coca leaf that causes all these behavioral effects, which he did. He succeeded. And he defended his thesis in 1860. So let's think about that for a minute. In 1860, Albert Niemann is the only person on the planet who is able to extract purified cocaine from the coca leaf. In 1861, Albert Niemann died under mysterious circumstances. Perhaps another cautionary tale, but that's the truth. So interestingly, one of the first people to systematically study the behavioral effects of cocaine was a young Sigmund Freud. So soon after the process for the purification of extraction and purification of cocaine was published, drug companies in Europe started producing it by the ton. The Merck drug company produced, tons and tons of cocaine. It was completely unregulated. So, if you're a physician, you could get it. If you were anybody, you could get it. And it wasn't all that expensive. So this was, when I say a young Sigmund Freud, he was 27 years old when he published this pamphlet called Über Coca, which translates into On Cocaine. And I say pamphlet, it's really not a book. The version of it I have is 19 pages long, and it's absolutely fascinating reading. Because Freud, one of the reasons Freud is still talked about today, because again, I'll try not to get on my soapbox, but he got almost everything he got wrong, right? But he could really write. I mean, you read this stuff. He was at least the English translations, he was a really a good and compelling writer. And this is really good reading if you ever get your hands on it, because of some of the stuff he says in there. Because he talked about the fact. Well, the reason he loved cocaine so much, and the reason he wrote this book, this little pamphlet that said all these wonderful things about cocaine, is because he was using tons of it. He didn't cop to how much he was really using, because he was a big time addict at the height of this.
How was he ingesting it? Just to be clear.
He almost everywhere you could ingest it, including injection. So he ate it, he snorted it, and when that wasn't working for him, he started injecting it. And which was interesting because he couldn't have done that much earlier, because the syringe wasn't developed until the 1860s. Anyway, for whatever that's worth. So, okay, so what else do I have to say about Freud? So, the interesting thing about this, 27 years old, probably the same age or the similar age as many of you, publishes this pamphlet telling people that cocaine is a wonder drug, which it really is, or was. Think about all the things. He identified the antidepressant, potential, mood enhancing, i.e. antidepressant qualities. It's the same mechanism of action as, many of antidepressant drugs today. I mean, same as the SSRIs and, the tricyclics, they're similar drugs. Just this one's got a much higher abuse liability, which he did not mention, even though he was becoming an addict himself. He got a lot of it wrong. He said it would be useful for digestive problems. He thought it would be useful for treating pain associated with menstruation, all these terrible things that he got wrong. But a couple of things he got really right, like the one I just mentioned. But what did he completely miss? So the incredible thing about this story, and there was a book recently published about it, which was that, cocaine, think about cocaine. It's the same class as procaine, but procaine didn't exist back then. So procaine is the local anesthetic you get when you go to the dentist office. There were no local anesthetics at that time, none. If you went in to have surgery, they had guys that held you down. They got you drunk and they held you down. That's all we had. And they could, we had opium, so we could give opium for the pain. That was it. We couldn't put, or, I don't know. I don't think chloroform was even around then. So etherdome was 1860.
I don't know how widely available it was.
It was early days at any rate. But cocaine, or Freud, he was a very ambitious guy. So, these caveats, I hear your caveats. But, this huge thing was, and he noted it himself in his own, he didn't put it in the pamphlet, but he noted that when he took it via the oral route, his gums became numb. He noted it. This was a huge discovery that this guy named Kohler, who was working in the building next door, wrote. He was an ophthalmologist. He wrote about it, used it for eye surgery, and became incredibly famous. In all the history books now, it's Kohler. Kohler and cocaine, local anesthesia, and led to all these local anesthetics we have now. Let me just tell you, Freud was pissed off. I mean, he completely missed it, had to sit there and watch, because he was not doing well. This is a very, very competitive place in Vienna at that time. Kohler becomes famous, and he completely misses it, does not put it in his little book. And moreover, everybody blames him, because what happens, this little pamphlet is widely read, leads to the first gigantic cocaine addiction pandemic, both in Europe and the United States of America, and they're all pointing their finger at Freud. How does he then go on to become the father of psychiatry? Can you imagine at your age if you screwed up that bad? Or if my age, if I screwed up that bad, nobody'd ever give us NIH money ever again or read anything we said. So anyway, that's enough about Freud. But the thing about Freud is this little book was widely read, and it led to a great appreciation of the pharmacological effects of cocaine. And again, cocaine was completely unregulated. So people in those days that were trying to make a buck, they would do things like put cocaine in toothache drops. As we just said, that's perfectly reasonable. We do much the same today, but we don't use cocaine anymore. We use procaine and lidocaine, drugs that don't have an abuse liability. Some other uses might have been a little more dubious. I don't think cocaine has much effect on dandruff, but it was sold for that purpose, and other ones are downright dangerous. For example, they combined coca with wine. So and this became very, very popular. So here's a couple of formulations: Peruvian wine of coca, coca of the Incas, and so the French folks. We lost one French folk, so please forgive me for what I'm about to say. So this is Bon Mariani. Did I come close? Bon. I'm from Kentucky. I can't speak French. So this is Mariani wine. Bon means wine. So Mariani was this Italian guy who had who'd moved to Paris again to make his fortune. So what Mariani did is, he decides to sell wine, and that's not good enough for Mariani. He's got to put another highly addicting substance in there. He put the coca leaves in there, so it's not cocaine. He took these vats of wine and put the coca leaves in there and let the cocaine leach out. That wasn't good enough for Mariani either. So what Mariani did, he sat down, he scratched his head, he's like, how can I get people to buy this stuff? So what he did is he sat down and he wrote out a list of what he thought were the 1,000 most famous people in the world at that time. This is the late 1800s. 1,000 people, and he systematically sent out 1,000 cases of von Mariani, one to each of those people. Waited a month or so, or however long things took back in those days, and he wrote each of them again. He says, I hope you've received my gift of von Mariani. Hope you've had a chance to sample it, and if you liked it, maybe you can write back and tell me what you thought. He received hundreds of these testimonials back, and he proceeded to use all these testimonials in his advertising. The ones he got were amazing. This is a partial list. The President of the United States wrote it. The King of Greece, Queen Victoria we mentioned, the Shah of Persia. What's the Shah of Persia doing drinking wine, much less wine with coca in it? Thomas Edison, Sarah Bernhardt, the most famous actress of the day, Jules Verne. There's Mariani. This guy is the King of Spain. This guy, I can't remember his name, but he was a French balloonist who was very famous at the time. So this list is incredibly interesting. Looking at all these advertisements it's really kind of comical. But this is all interesting. But by far, my favorite testimonial was the one written by His Holiness, Pope Leo XIII. And Pope Leo looks like he's happy about something. So he liked von Mariani so much that he awarded Mariani a gold medal in recognition of the benefits received from von Mariani, a tonic for body, brain, and nerves. So before today, maybe you heard about von Mariani, maybe you haven't. It's an interesting story. But even if you hadn't heard about this, you probably know that the coca and Coca-Cola right there, the word originally comes from the fact that coca leaves were put in the formulation for Coca-Cola. What you may not know is that von Mariani led directly, more or less, to the development of Coca-Cola. So this guy is John Pemberton, and John Pemberton ran a drugstore in the 1880s in Atlanta, Georgia. And so I don't know if you guys remember this, but well into the 20th century, where you went to get your soda was to the drugstore. So you went to the drugstore and they had soda fountains. They didn't come in cans and bottles. You went and, they called them soda jerks. This even went out to the 1950s, and that's where you got your soda from, like Coca-Cola. And it was actually kind of advertised as having some sort of quasi-medicinal property. So that's why you went to the drugstore to get it, even though basically it was just to quench your thirst. So what happened is Pemberton starts seeing, this von Mariani and like products end up in the United States. They start becoming very popular. So Pemberton decides to get in, jump on the bandwagon. So he comes up with exactly the same thing as von Mariani, steals the idea, but he calls it Pemberton's French Wine Coca. And he starts selling it in Atlanta, and it's very popular. Then something bad happened for John Pemberton. I come from the southern United States. The place that I was born, which is a gigantic county in Kentucky, still is a dry county. You cannot buy alcohol there in 2013. You have to drive all the way to Lexington, which is almost an hour and a half away, to buy beer, for God's sake, in 2013. So the South is still a strange place, and it was a strange place in the 1880s, and back in the 1880s, they outlawed alcohol in Atlanta, Georgia. This was a big problem for Pemberton because he had to take, the coca was fine, but he had to take the alcohol out, okay? So he had to take the alcohol out of his product. He replaced the wine with carbonated water and cola nuts, left the coca in, and called it Coca-Cola. So this was good for Pemberton. So the new product, Coca-Cola, sold very well. He advertised it. Here's some of the advertisements. You could get it for a nickel back in those days. Things went along great until 1918 when the Harrison Narcotic Act came in, and darn wouldn't you know it, now cocaine's illegal. So he's got to take the coca out. Pemberton didn't like this at all. So he'd been making money hand over fist selling Coca-Cola. And he really thought, so again, he would take these vats of Coca-Cola, and he didn't put cocaine in there, he put coca leaves in there, okay? And then the cocaine would just leach out. So who knows how much cocaine was ending up in there. But a lot of other stuff was ending up in there too. He thought this was all really important, okay? You know, cocaine's a little bitter. He thought this little bitter flavor of Coca-Cola was really important. So he scratches his head. And back in those days, I mean, someone's got to be creating this stuff after it becomes illegal. And unlike marijuana, which is Schedule 1 in 2013, cocaine is Schedule 2. Okay? It's still used as a local anesthetic in 2013. If it's used legally as a local anesthetic in 2013, someone's got to make it. And there's a drug company in southern New Jersey. It's the only company in the United States of America that is licensed to produce cocaine for medicinal purposes. That same company has been making medicinal cocaine for over 100 years. That's the company that John Pemberton called and said, I got a question for you. When you make this cocaine, what do you do with everything that's left? And they said, we throw it out. He says, why don't you give it to me? So they gave it to John Pemberton. He put it in the Coca-Cola in 1918, and they put it in the Coca-Cola in 2013. There are still coca extracts in the Coca-Cola you are drinking right now, and that's the truth. Woo, that was a lot on fun and games. No time for anything else. But the rest of the story does go quickly. So there are, relatively quickly, so there are over 2 million regular cocaine users in the United States of America, and this is where they live. I find this map fascinating. So red is high, white is actually in the middle, and blue is low. So let's look at Long Island. I mean, Long Island's in the middle. New York City is in the middle. Philadelphia, where I live, not that bad. Where Anto is going, northwestern Vermont. What's going on in northwestern Vermont? I thought it was just a bunch of stoners up there. What's going on? Do you know?
No, nothing. That's the problem. They're
Well,
bored. It's like the no enrichment.
but how do they get it up there? But then there's other things that are interesting. So I can look at this forever. So the thing that you notice are college towns and state capitals. So, I mean, Miami is a port. Tallahassee's not a port. It's a college town. New Orleans is a port. This is Austin. That's Nashville. That's Columbus, Ohio. All of those are state capitals and college towns. I've given this talk in Arizona and Colorado. They were shocked. I mean, what's going on in Colorado? I mean, why is so much cocaine being consumed in Colorado? Maybe it'll be interesting to see if the marijuana thing causes it to go down. So anyway, I think this is fascinating, but I've spent enough time talking about things that I shouldn't be talking about. So animal model of cocaine addiction. So we study rats and mice. As I said previously, mainly rats. The people in my lab have convinced me to study mice, and they've regretted it, that they've gone down that road. I much prefer this little critter. Rats are nice. So this is IV self-administration. I assume if you guys know what progressive ratio is, you know what this is. Progressive ratio was Elliot Wallenstein, 1963. Kodos was the first author, but Elliot's still around at the University of Michigan. He writes popular books now. They're really good. So what happened in 2006? So in 2006, Ghazaleh Sadri Bakili called me. So I was in my office at Boston University, and she was my former graduate student. She was then a junior faculty member at Harvard. She calls me up and she says, did you see this RFA about epigenetics and addiction? And I said, no. And she said, well, that's what I do now. Would you be interested in, maybe putting together a project and putting in a grant on the epigenetics of addiction? I said, sure, and hung up the phone and opened up Google and Googled epigenetics to see what the hell I had just gotten myself into. This is the truth. So, so I figured out the first thing we needed to do was find an mRNA to change. So this is my definition. This is the new definition of epigenetics. So the first issue was if we're going to chase down these epigenetic modifications in adult animals, that we had to find an mRNA that really changed. Okay, this is not transgenerational. I'm like Emma Whitelaw here. Okay, we're starting at the beginning. We're going to talk about transgenerational in 10 or 15 minutes. So the first thing we needed to do is find an mRNA that changed. So that's Ghazaleh there and there. And so we just decided to do 14 days of cocaine self-administration, which David will tell us is not enough, but that's 2 hours a day for 14 days. Whatever changes are taking place in the brain, wherever they're addiction-related or not. This is a lot of cocaine these guys take. Then we just gave them a week off, make sure whatever change we saw was persistent. And the controls were yolk saline animals. So we killed the animal. We took out all the spots in the brain, the normal suspects, the nucleus accumbens, prefrontal cortex, amygdala, hippocampus, BTA, et cetera, et cetera. So we took all these areas out. And the first thing that occurred to us is, well, let's do gene shift. Okay, for various, back then, gene chip. It would be an expensive experiment to do now. Back then, it was basically prohibitive. It was a lot cheaper for us to just do real-time PCR on the usual suspects. So we just looked at the transcripts, the things that we had in the freezer, dopamine receptors, enkephalin, dynorphin, CFOS, BDNF, etc. And we just did a ton of PCRs. So what I'm showing here on the next slide is protein. But this protein finding was, I just don't want to talk about all the mRNAs. Like there was a lot of them. A lot of mRNAs in a lot of different areas. The area of the brain that we typically study is the nucleus accumbens, but the biggest change, and I wanted to chase down the biggest change that we saw of anything was BDNF, mRNA, and protein in the whole of the prefrontal cortex. Okay, and this is a big change. So and there was an equivalent mRNA change. Okay, so then we wanted to look at the epigenetic modifications that might generate this change in protein. Okay, so just as you guys all know, BDNF is brain-derived neurotrophic factor. A whole bunch of studies have shown that BDNF plays a role in plasticity generally, and cocaine-induced and drugs of abuse-induced plasticity in particular. And so, there's got to be there's going to be some changes in acetylation, methylation, etc. that are going to be driving these changes in transcription. At least that was our hypothesis. Okay, so a little bit about BDNF. BDNF is a really curious critter. So BDNF has 8 promoters that drive 8 exons. So, what little I know about this says to me that because these 8 exons generate exactly the same protein, that this must have evolved 8 different times throughout, in the last billion years or so, right? So you've got 8 different forms of evolution, all of which were conserved in us mammals. But they all produce the same protein. So BDNF must be really important for a whole lot of different species. So it's really interesting. So there's, when we started these studies, there were 7 exons. In the middle of this study, they discovered 2 more. Thanks a lot, guys. So now there's 9. And so the first thing we did is like, okay, well, which exons are driving our change in protein? So we looked at these. So this is the new exon 4, which is the old exon 3. So a paper from 2005 is going to call it exon 3, a paper from 2013, or this was from 2010, is going to call it exon 4. By far the most widely studied are exon 1 and exon 4, and of those 2, exon 4. So like the learning and memory literature, they really have focused on changes and seen changes in exon 4, and correspondingly, that's what we saw. We saw no exon 2 expression at all, and we saw a little bit of a trend for an increase in exon 1, but a nice big increase, commensurate with the change in protein we saw for exon 4. So, the conclusion is this change in exon 4 specifically that's driving our change in protein that we see in the prefrontal cortex. All right, so the next experiments are, so this whole field of epigenetics, narrowly defined, has to do with this assay called the chromatin immunoprecipitation assay. So what this allows you to do is look at the association of chromatin elements with promoters. And so like in this case, so you've got Euchromatin and Heterochromatin, and you've got this is a histone acetyltransferase. So this transfers acyl groups on here, which opens up the chromatin, right? So and when it's open, we're ready for transcription. And how we can study these associations specifically between transcription factors and the things that they transcribe is this CHIP assay, chromatin immunoprecipitation. So I don't want to get into all the details of this, but it's just basically a co-IP, you reverse the cross-links, and you do PCR. Which sounds really simple, right? And conceptually it is. I can just tell you that if you sit down to do it in the lab, it's not so simple. Making this work is tricky. So the folks that have this, it takes a long time to get good at it, but once you're good at it, you're golden. At least 5 years ago, you could really get a job just because you could do a CHIP assay. Okay, so we did these CHIP assays to see, and you got to have a good antibody. So you have, the reason we looked at H3 is because the H3 antibodies are the best of all the histone-associated things. So there's an acetylated H3, and we're looking at its association with all these BDNF promoters. And we showed that after cocaine, relative to these saline animals, we showed an increased association of acetylated H3 with BDNF exon 4. So that tells us that we've got the open chromatin state, and once the chromatin's open, that the acetylation's coming in and acetylating H3, which is opening up the chromatin. And once the chromatin's open, then the transcription factors can come in and bind. Okay, so what are the transcription factors for BDNF? there's more than this, but these are the big ones. And by far the most widely studied are CREB and MECP2, which you'll remember from half an hour ago or whatever, were the ones that I highlighted at the end of our epigenetics talk. So we've got, it's acetylated, now the transcription factors can come in, but CREB and MECP2 do opposite things. So as you guys all know, CREB drives transcription. So if CREB comes in and binds, it drives transcription of various things like CFOS, etc. Whereas MEC, and it's in green, telling you that it's driving the transcription, whereas MECP2 is in red, because in most cases, MECP2 is a transcriptional repressor. So MECP2 comes in and it shuts down transcription. Okay, so this is go and that's stop. So we wanted to look at both of them, using again the CHIP assay. So we looked at the association of phospho-CREB with all these exons, and we showed an increased association of phospho-CREB with BDNF exon 4. So again, you know, CREB is driving transcription, and specifically, it's driving transcription of BDNF exon 4, just like we showed before. So this is consistent. Whereas there's a decreased association of MECP2. So whereas previously, so what CREB is doing is like putting your foot on the gas. Okay. What MECP2 here is doing, we have a decreased association of this. So this is equivalent to taking your foot off the brake. Okay. And these two processes together drive the transcription of the BDNF protein, specifically via exon 4. Okay, so this paper, which we published in 2010, showed increased association of acetylated H3 with BDNF exon 4, opens up the chromatin. Once the chromatin's open, the pCREB gets in there, drives transcription of BDNF, and the loss of MECP2 is, you know, is taking a break off of driving the BDNF protein.
No question.
Yeah.
How close are the binding sites on exon 4?
Yeah, good question. I have no idea. They're not that close, but I don't know how far apart they are. It's a huge gene. It's really big. Any other questions?
Yeah, so in specific cell types, do they use specific exon? Is there a specific exon used like microglia use one exon versus neuronal BDNF expression? You know?
That's a good question. I don't really know. I know that, well, at least in many, I mean, in the neurons that work, the same neurons can have the same exons do the same thing. So I know that there's cases for like, there's many cases where 1 and 4 are in the same neuron and both driving transcription of the same protein. So, you know, they're all differentially regulated. So the upstream processes of 1 and 4 are all different. So like, you know, what is it? CAMK4 drives exon 4, for example, but not exon 1. So it can do the same, it can do different things in the same cell via different signaling mechanisms.
Okay.
But I don't really know the extent to which there's differential distribution of these exons. I'm sure they are throughout the brain and certainly throughout the body, but even within the brain. Oh, we're not done with this story. Okay, so big deal. Does it matter? So do these changes in BDNF transcription make any difference? So Carolyn Bass worked at Children's Hospital in Boston back then. She now is at the University of Buffalo. She created a adenoviral shRNA targeting exon 4 of BDNF, so we can knock down BDNF in the prefrontal cortex, and it really worked well. So we got a 60% reduction. So if you guys haven't worked with viruses, 60% is really good, and it also produces EGFP. I'm really glad I don't have to explain a progressive ratio to you, because then we put the guys, so we did an acquisition of cocaine self-administration, between these two groups, the control and the knockdown of BDNF in the prefrontal cortex. The acquisition was the same. Then we switched them over to a progressive ratio, and to our surprise, the guys that had reduced BDNF transcription in the prefrontal cortex really worked to get cocaine. Really worked. They didn't press as much as David's animals. Our curve is not that steep. It's more like this. But I mean, we're talking, in order to go from the 16th to the 17th infusion, we're talking 400 presses to get, you know, to get the next infusion of cocaine. So they worked a lot harder. So these results indicate that cocaine-induced increases in prefrontal cortical BDNF transcription represent a compensatory adaptation that reduces cocaine reinforcement. Okay. All right, so back to epigenetics. So at the beginning, so does anybody have any questions so far? So at the beginning of this part of the talk, or the middle part of the talk, I left this word out, this word inherited. So I can tell you the part of the talk, I've been giving this middle things, that this whole story about these epigenetic marks. You know, I've been doing this for 5 or 6 years. And woe unto me, I actually went to a very molecularly oriented department, gave this talk, and holy crap, they jumped all over me. And they were concerned, I'll get to you in a second. So their concern was, I was like, you know, I mean, you're jumping on me. Like, I told you I'm not a molecular biologist. I mean, why are you so mad at me? They were mad at my use of the word epigenetics. They were, I mean, their thing was, it was semantics. They were like, you don't even know what epigenetics is. None of this stuff, you keep saying epigenetics, none of this stuff you've talked about is epigenetics. I mean, this guy was really mad at me. I'm like, finally it sunk into me that it was not heritable. The changes that I just talked about, none of this was inherited. This was just changes in acetylation and methylation. The modern definition of epigenetics, I will argue, encompasses heritable changes, but also encompasses changes in an adult organism that involve epigenetic marks like acetylation and methylation. But man, some folks, one of the original definitions was that that epigenetics means it's heritable. And if it ain't heritable, it ain't epigenetics, so you don't know what you're talking about. And there's someone at Penn, who shall remain nameless, who falls in this category, and she thinks I'm the biggest idiot in the world. And the next paper I'm going to talk about pissed her off royally because I got epigenetics in the title. She didn't like it at all. But I'll note that, well, where was the title? My other title, well, it was chromatin remodeling, you'll remember. So it's cocaine-induced changes in chromatin remodeling because one of the reviewers, we had epigenetic modifications in the title before, and the reviewer made us take it out. Made us take the word, we had to take the word epigenetic out of the entire paper. Question?
Yeah, I was just thinking about BDNF and drug addiction. From my understanding, behaviors like exercise that increase BDNF also attenuate drug addiction and I guess self-administration, but it's also associated with neurogenesis. So is it the increase in BDNF that's acting directly on reward systems, or is it the neurogenesis and all those things, or is it?
Well, there's not much neurogenesis. So the neurogenesis happens in places like the hippocampus, which might influence addiction. This is prefrontal cortex. There's little or none happening there. But the interesting point here is it has to do with where the BDNF is, because like Yabin Shaham's work, you know, with their incubation phenomenon is positively correlated with BDNF levels in the VTA, which is the opposite of what we are saying in the prefrontal cortex. So, you know, BDNF, depending on what part of the brain it is, I mean, again, we were surprised at this. This is not, the reason I think it's right, number one, because we replicated it, and number two is the exact opposite of what we were expecting. So I think it must be right. But it's also the opposite of the effects of BDNF in other parts of the brain. Hey, David, where's that picture taken? Close. It was after the Arcachon meeting, but that's Bordeaux.
Is there?
Yeah, it is.
Yeah, I'm saying to go on. Okay.
So he's referring, there was this meeting. These guys talked me into going to that meeting in Arcachon, which is a smart, because I was a little miffed because they didn't invite me to come and give a talk. But so I was like, I'm not going. They didn't invite me, so I'm not going.
Not really. I was 200 kilometers from there, and I wasn't invited either.
Well, they invited Clivus. They invited Clivus, my former mentor, and they invited Marina Wolf. They actually apologized to me when I actually went. They were like, we just had too many glutamate people. And I was like, I'm fine, it's good. But these two were like, you got to be kidding me. They're having a meeting in Arcachon in France, and there was so much good wine, so much good food. I don't even remember half that meeting. But this is after the meeting. Then we went and visited Serge Ahmed in Bordeaux. So the bottom line of this is come work in my lab, and we have fun. We go nice, do paintball, go to Bordeaux. So this is Lisa Briand, who's a postdoc in the lab. She's now a research assistant professor at Penn. This is Farah Vassler, who at the time was a graduate student. As I said, she's the star of this next part of the story. So going back to this word inherited. So I'd been jumped on about this heritable thing. I was like, okay, I promise never to say epigenetics again, and then lied and actually did it. But I hadn't really read this literature, knowing, I mean, all this stuff about transgenerational, you know, now there's a big literature about exposure to toxins and transgenerational effects of toxins. Tracy Bale from my place at Penn, she does high-fat diet. She starves the animals, sees all these transgenerational changes. So the seed was planted, and Fair came to me and she wanted, you know, it's like, but there was no way I was going to assign this experiment to someone. So the notion of doing transgenerational effects with addiction, because it was going to be hard, it was going to be expensive, it was going to be time consuming, and it probably wasn't going to work. Okay, so but Fair came to me, and her thesis was going along, skipping right along. She had a couple of really nice papers already. Came in my office, said she wanted to do it. I tried to talk her out of it, told her that she was going to delay her thesis. You know, it was going to blow up in her face. It was going to be bad. Well, she didn't listen to me. She did it anyway, and that was smart. So what did Fair do? So she did 60 days of cocaine self-administration or yolk saline. Why did we do 60 days of cocaine self-administration? Goodness help us, that's how long a sperm lives. Okay, so your sperm, they turn over every 60 days. Okay, they're born all the time, but 60 days they're born, they live, they die in 60 days. Unfortunate for us, because now we're stuck with 2 months. I don't like these long, David doesn't like to do these long experiments either. I certainly don't like to do them. It takes too damn long. So we did 60 days of cocaine self-administration. This is the males, okay, just the dads. So and you can see from the first part of the talk, you know why we did dads and not moms, okay? Because we only had to go to F2 rather than going out to F3. It's a lot easier. So then we took these dads and we bred them with naive females. The dads went away, never to be seen from again, just like real cocaine addicts in real life. And then we let the progeny grow up, and we asked them what they thought of cocaine, if they liked cocaine. So the F0 generation are the moms and the dads. So these guys took cocaine, and every time these guys took cocaine, these guys got a shot of saline, and then they bred with these nice little girls here, and they got pregnant, and these guys went away. And then, so what we call these guys, so the offspring are the next generation, these guys are cocaine sired, and these guys are saline sired. So we asked the female offspring, these are 90-day-old animals, we asked them, what do they think of cocaine? So this is the acquisition of cocaine self-administration. So this is a moderate and high dose of cocaine. You can see the acquisition curves are exactly on top of each other. Contrast that with the males, is that we have delayed acquisition with this lower dose, but they actually catch up. But here at the higher dose, we have delayed acquisition and reduced maintenance of cocaine self-administration. So again, everything about this project has been the opposite of what we expected. Our expectation would be, number one, my expectation would be nothing, what we saw with the females. And if I thought something was going to be happening, I thought they would like cocaine more. But they liked cocaine less. Or at least this appeared that they took less. The next question was, why did they take less? Okay, but there's no question they took less cocaine, which was a surprise to us. All right, so there's delayed acquisition of cocaine self-administration in the male but not the female progeny of cocaine-exposed fathers. Why? Well, the first question was, are they stupid? No laugh for that? Come on. So we did acquisition. These are old data. These are not the data that finally ended up in the paper, but we did acquisition of food self-administration, and the curves were right on top of each other. And we've done a whole bunch of other learning and memory tests, and learning and memory is not impaired in these animals, so they do not appear to be stupid. Is cocaine less reinforcing? So then the question, so these are the very same animals we did the acquisition on. So once the acquisition was over, we shifted them over to progressive ratio. We asked them how hard were they willing to work to receive cocaine. So the females, not surprisingly, were exactly the same. So their acquisition curves were the same. Their progressive ratio breakpoints were the same. So these guys, remember these low doses of cocaine among the boys, so pink girls, blue boys, aren't I clever? So they eventually caught up, and when we shifted them to progressive ratio, they were exactly the same. These high dose guys, where the curves stayed like this, when we shifted them to PR, these guys worked, the cocaine-sired guys were not willing to work very hard to receive cocaine at all, relative to the control animals. So the bottom line was the male offspring of cocaine-exposed sires would not work as hard to receive cocaine. So the next question was, so this harks back to, so these animals don't like cocaine so much. So what was the first thing that went through our mind? BDNF in the prefrontal cortex. So just to remind you, what we showed in that 2010 paper was that we had a compensatory adaptation in BDNF in the prefrontal cortex. So cocaine self-administration increased BDNF, mRNA, and protein in the medial prefrontal cortex. And that, as I just said, is a compensatory mechanism that decreases the reinforcing efficacy of cocaine, which we previously studied the same way using a progressive ratio schedule. So our hypothesis was that maybe these animals had increased BDNF in their prefrontal cortex, in mRNA and protein in the males, but not the females. And obviously that's what we saw, or I wouldn't be talking about it. So this bar here should be pink. So we looked at the protein levels in the males and the females. So there was no change in phenotype in the females, no change in BDNF levels. We had a change in phenotype in the males. We saw a nice increase in BDNF in the prefrontal cortex. We looked at the exon specificity. We see this nice change in exon, BDNF exon 4. And then we did the CHIP assay where we looked at acytylated H3 association with these BDNF promoters, and the exact same one popped up again. So we had increased acetylation of exon of H3 associated with BDNF promoter 4. So these male cocaine-sired rats have increased mRNA and protein in the prefrontal cortex, which may decrease the reinforcing efficacy or cocaine reinforcement. So in other words, bottom line, these guys like it less, maybe because there's increased BDNF in the prefrontal cortex. So the next thing, if you see a change in phenotype, you always have to confirm it. And unfortunately, BDNF is tough to study. There are no good drugs I mean, that's why we had to do shRNA in the previous study. We didn't want to do that in this experiment for various reasons. The drugs are terrible. So TrkB is the receptor that BDNF signals through. There's this drug called ANA12, which is not water soluble, which is a big problem for us because we would have liked to put it in the prefrontal cortex, but we can't. It's like you got to have 90% DMSO to get this stuff to go into solution. It's just a bad drug. But we did the best we could. We administered it systemically during the acquisition of cocaine self-administration among saline-sired and cocaine-sired animals. Okay, so this is the totals of the first 10 days of self-administration. So this is a replication of the effect we saw before. This is the acquisition of cocaine self-administration at the high dose among the saline-sired animals. That's decreased in the cocaine-sired animals. When we take the cocaine-sired animals and gave them this TrkB antagonist, we reversed the phenotype, which is our expectation. So this demonstrates I mean, this is not a perfect experiment, but it's consistent with our hypothesis that it's BDNF in the prefrontal cortex that's driving this behavior. Okay, so then we get to the critical question. So what's the mechanism of cocaine-associated information transmitting from father to son? So you guys already know what the answer is going to be, and it's sperm. So this is tomography of sperm. These are basically pretty pictures. But you can see these are saline animals and cocaine animals, so if you just look at them, you can see they're different. So these are tubules, testicular tubules, and this is acytylated H3 levels, and that's a nuclear dye. So these are pretty pictures. But we looked at acytylated H3 in the histones. Well, let's not get into it. There's histones and protamines, but let's not talk about all that stuff. Histones are important, and they can be acytylated in the sperm. And we saw increases in acetylation of H3 in the sperm of the cocaine-sired dads, but not the saline-sired dads, or at least a difference between the two. We did a CHIP assay on sperm. Trust me, I know way more about sperm than I ever wanted to know, and we keep having to do experiments like this, and it really makes my head hurt. But we did it, and it actually worked. It was all the promoters. So again, talk about not a perfect experiment. This is the sperm. This is, there's no way of knowing this BDNF, it's consistent with our hypothesis, but it's all the sperm. Like it could be BDNF that's going to go to the liver, whatever. There's no indication that there's no way we can do an experiment to say that this change in the sperm is going to be specific for the BDNF and the offspring. But we did the best we could, and the data were consistent. Okay.
Do you know if just a single exposure of cocaine alters acetylation in the sperm? Have you guys done those experiments?
We did. We just gave it to them systemically, which is not the same thing at all, but it didn't produce a change.
And you don't know how long-lasting that change is?
Correct. We don't. I really don't want to do any more sperm experiments, but I
It has
have
to be
to.
self-administration. It doesn't have to be just non-contingent.
Well, that's what we did. The experiment to see if an acute injection was just a single shot, but it's not.
You haven't done it for a long time.
No, we haven't.
Non-contingent phenomenon.
Well, then another experiment was, we could take sperm and put cocaine on it. Just take a sperm in a dish and put cocaine on it. It's like, trust me, there's all these experiments I don't want to do, which would be good experiments.
Because it has clinical implications. Once a crackhead, always a crackhead, or can you recover and then your children are not altered?
So the interesting issues about all this stuff about sperm that I don't want to know is, since you asked, it's 5 o'clock, but you're asking questions. So just remember, if I give long-winded answers, it's your fault. So way back in the 70s, they did these experiments where they took tritiated cocaine and they gave it to rats. These are nutty experiments. So they gave the tritiated cocaine, let it distribute across the tissues. They froze the animals and they cut them longitudinally, the whole animal. And the number one place where the tritiated cocaine ended up, not surprisingly, was in the brain. Number 2, the testes. And there are no transporters in the testes, none. So there's no dopamine, norepinephrine, serotonin transporters. We know that. They know that there's a protein. Those guys back in the 70s, it was not easy to do things like sequencing back then. They characterized a protein, they know it exists, it binds cocaine, nobody knows what it is. So I keep telling postdocs in the lab, hey, you should figure out how to characterize that protein, but unlike FAIR, nobody's bit on that yet. But that is interesting. So there's something down there that may be influencing the sperm, and which may be part of the reason that we see it in the male line, may not see it on the female line. Okay, so the bottom line of all this, and I am almost done. This is my next to last slide. 3 more slides. So delayed acquisition of cocaine self-administration. Operant learning was fine, so they're not stupid. Increased feeding of protein and mRNA in the prefrontal cortex, which may reduce cocaine reinforcement. The TrkB inhibitor normalized the behavior, which is a nice reversal of the phenotype. And this at least is suggestive that there may be an epigenetic change in the BDNF promoter in the sperm. So here's the paper, which was published in January of this year, and I was so pleased I got the word epigenetic into the title. But is it epigenetic? Everything that I've told you, why was it important for me to get this word in there? Well, it was important for me to get this word in there so people would cite it. That was the real reason. But why did I convince myself it was epigenetic? Because is this intergenerational or transgenerational? This is intergenerational, okay? So I also got away with not, I thought the first thing the reviewers were going to say is, okay, that's all fine, but you got to look in F2, which they didn't. I can't believe it. We've looked at F2. It looks like the phenotype isn't there. It's hard to tell, but at least the initial evidence is the phenotype doesn't make it to F2. But to us, it's important, because again, the notion that whatever we've done, like all the alcohol I've drunk, all the marijuana that some of us might have smoked in college. What effects might these have? Thank God I'm not having any more kids. Like all the things that I've done that may be visited on them. I've got a reasonably healthy child, thank goodness. But all these things that might happen, even if it's one generation, and happily the editors of Nature Neuroscience agreed with us, that the epigenetic change being the change in the sperm, so it is a modification in the sperm, it's acetylation, which narrowly defined, it doesn't have to be transgenerational, the way we define it. So it's an epigenetic alteration in the sperm that changes the phenotype in the offspring, which we thought was important enough to go into a journal like this. And happily they agreed. That doesn't mean there aren't, that's the critical unanswered question. What happens in the maternal line, which is going to be very hard to answer, but we're working on. What happens in F2, we're working on this. As you can imagine, it takes 6 months to generate these animals, 6 months. So these are tough experiments. So anyway, those are the guys. Fair is the star of the second part of the story. Gosley is the star of the first part of the story. Heath and Sam made huge contributions. That's the rest of the people in the lab who all contributed one way or the other. NIDA paid for it all. Thanks, guys. Thanks, Nora, who was here yesterday. And thanks to you for sticking with me till 5:05. And if you have any more questions, I would be happy to answer.
Are there any other explanations?
Any other explanations?
Besides the transfer of the epigenetics to the male?
So the things that you learn about that I never would have thought. So one of the things that we had poor fare, one of the things that I made her address before we submitted this paper is that this person named Frances Champagne, who's at Columbia, who does terrific work, and she's done a lot of this transgenerational work. She worked with Michael Meany, who's one of the big figures. But she's in a psychology department. So what her notion is that the female, let's make sure I can get this right, that the female selects the male. Okay, so that based on that the female has some idea about the fitness of the males and will choose out in, ethologically will make these choices. So the idea epigenetically being that the female can tell if it's a dud, she's got a dud. And in this case, the cocaine guy would be the dud, and the saline guy would be the strapping good guy. Okay, and that her behavior will be altered such that if the dad was a dud, she's going to not care about her kids. Whereas if the dad's a good dad, seems like he's going to be a good dad, she'll give them extra attention. And the thing that Frances studies is licking and grooming, which is again a thing that I never poor favor. She had to do these experiments because that was another thing I thought for sure they were going to make us do it. So the notion being that the maternal behavior would be influenced just by the female, even though they were only together for like a day. That being exposed to this guy who impregnated her was going to influence her maternal behavior. So we assessed all these maternal behaviors and showed there were no difference. So we had to do that. So that was a question asked and answered. But other than that, that's another big thing that you run across in the literature about an indirect mechanism that you can get to a phenotype. I can't think of it. Can anybody else think of any others?
Well, I mean, you're getting big changes because you've got all that immunocytic chemistry change on the acetylation.
Mhm.
Yeah. Okay. So it looked like there was changes on lots and lots of genes, presumably, or is it just one gene?
We only looked at one. So
What about your immunocytic chemistry? Was that just with one? So that's just looking at one. So I was just wondering if because you could have repressors or something changing, so you've got difference in the efficacy of I don't know the histones dissociation or something like this during. I was just wondering if there are lots of other explanations besides the actual epigenetic phenotype going through.
So I think it must. So my guess is it's no matter. I mean, we were lucky because we had a phenotype and we had we'd previously done this paper looking at this epigenetic modification. The short answer is I think it's going to be an epigenetic modification of whatever. BDNF is obviously not the be-all end-all of addiction or anything else. I mean, there's a lot of other genes that are undoubtedly involved. We evidently got lucky with the one we chose, and we specifically chose to have very targeted experiments. Now we're taking a step back. I mean, we're right now spending a god-awful amount of money sequencing the genome. We're doing me-dip experiments looking at methylation and acetylation of sperm. I mean, it's costing us like $30,000. And we're going to actually, sequence all, some of the epigenetic modifications in sperm and look at the entire genome. So now we're taking a step back, and Katie bar the door, and whatever comes through, will be there. And with our luck, I'm going to spend all that money and nothing, including BDNF, will come out.
So I guess cocaine addicts have weird sperm.
Evidently.
Yeah.
Which we don't know from. There is some evidence from animals that they have weird sperm, other than our data. Not methylation, but the sperm is weird, and that's another question I get is is the sperm damaged? And there's it's inconsistent. Some people see no change. It's actually been looked at. Some people see changes, some people don't.
Sizes between your saline and your control and your
So we called them all the 10.
How do you call them?
We did not cross-foster. So thank goodness I talked to Eric, who, David Deep, they spent over $20,000 doing not cross-fostering, cross-gestation, right? They did cross-gestation. David did the cross-gestation. Was it cross-fostering?
Cross-gestation. The problem is that when the mother were, when you send the sperm, they get the best mom. The men that are going to be super nurturing. So it's also biased if in your experiment, the mom were in direct contact with the male. Whereas when you send the sperm, they always have very good mom that will take care of all the pups the same way. So that's also that was the issue.
So cross-fostering is an experiment. That's an important experiment to do, which we didn't want to do and we didn't do. So Eric did in vitro. It was actually in vitro fertilization. Cost a fortune. and it wiped out their effect. Poor David. So yeah, thank goodness we didn't go down that road too. I mean, we're spending way too much money on this as it is. I don't have Eric's kind of money. Tell him that tomorrow. No, don't tell him that tomorrow.
Okay, Chris, thanks.
Thanks, Eric.